Ladies and gentlemen, if I could have your attention. Thank you so much. So a very warm welcome to the annual Forum lecture. If I could introduce myself briefly, I'm Robert Lechler. I have the privilege of being the president of the Academy of Medical Sciences. And for those of you that don't know the Academy, we're one of 4 national academies. We're the youngest, the most interesting, the most dynamic, and the best. So we've been in existence for 20 years, and we have a very simple purpose, and that is to promote biomedical and health research and its translation into benefit for patients and the population. Thank you very much for being here and dragging yourself out of the beautiful weather outside. Just in terms of housekeeping, can I ask you to be sure that your mobile phone is at least silent, preferably off, so you have no distractions? That's up to you, but certainly silent. Of course, what I'm about to say is incompatible with turning it off. If you wish to tweet, then use the hashtag Forum2019. The Twitter details will be on the holding slide for reference as we go. And the audience, reminding the audience the lecture is being filmed, and the report and recordings will be made available in due course following the event. Now, you may ask, what is the Forum? So let me introduce that to you. It reflects one of the 6 things when I explain what the Academy does, one of the 6 things the Academy does, which is attempts to foster closer interactions and partnership between 3 sectors, academia, NHS, and industry, which I hope you'd agree, if we want to maximize the benefit of our assets and we want to accelerate the translation of discovery into patient benefit, those 3 sectors really need to work together. So one of my predecessors had the foresight way back in 2003, very early in the Academy's life, to set up this thing called the Forum, which has just gathered momentum over the years. It now is made up of 41 member organizations that are a mixture of industries, academic institutions, regulators, and the like. The joining fee is very modest, so if you're representing an organization which is not part of the Forum and you think you'd like to be, don't hesitate to get your checkbook out before you leave and talk to one of the Academy staff, and we'd be delighted to consider having you as part of this family. What the Forum does, apart from this annual showcase event, is we organize workshops and roundtable discussions on topical subjects of interest to those 3 sectors, things to do with clinical trials, regulation, even drug pricing, all those sorts of things that can be obstacles to delivering some of the things that we all want to deliver. So on this slide, you'll see down the right-hand side examples of things that we've been doing recently. So there you are: adaptive trials, future of gene therapies, training, the annual lecture, mechanisms underlying multimorbidity. So a really interesting suite of topics. So as I say, quite seriously, if you represent an organization that isn't part of the Forum, then do talk to us about joining. So let's get on to the main business of today. And I'm very excited about this afternoon's program. The Forum Annual Lecture is a very prestigious event, and we're delighted to have someone highly appropriate to give the plenary talk. The theme being the promise of human genome editing for rare and genetic diseases. Now, it's become a very topical subject, and you'll be aware of that, and I guess that's why you're here. But actually, the concept of gene editing has been around for a while, and there are some predecessor technologies like zinc finger nucleases and TALENs that have been around for several decades, actually. And in fact, CRISPR itself was discovered in late 80s in Japan. So even that, as an entity, has been known about for some time, but its real potential, I think, came to light in the early 2010s, and of course led to some very famous patent filings, which continue to be a business that keeps some lawyers occupied right up to the present day. But it is an immensely important technology. If I'd known what was going on in the political climate, I might have had this talk on political editing rather than genome editing. We certainly need a bit of political editing, but maybe that's beyond the powers even of a company like Intellia. The design of the program is that we're going to have the plenary talk in a moment, and then we'll have some time for Q&A on that subject and the material that John will have presented. And then we'll have 4 shorter talks covering some aspects of science, some aspects of ethics related to this subject, and the patient voice. I'm really pleased that we have that included. And then we'll have an open general discussion, which I invite you all to participate in. So, without further ado, let me introduce our plenary speaker, Dr. John Leonard, who's the Chief Executive of Intellia Therapeutics. John had a distinguished and highly successful career for 30 years in pharmaceutical R&D, and then retired from that to take on this new challenge, initially as Chief Scientific Officer, and now as Chief Medical Officer, and now as Chief Executive of one of 3 companies that are really driving forward this technology towards patient application. So we're very fortunate that he's agreed to give this talk. I can't think of anybody better to give an overview of how this field, this very fast-moving field, is developing, and the huge impacts that I predict that it will have in a broad range of medical disciplines. So, John, welcome. Thank you very much for coming.
So thank you, and I'm certainly pleased to join you this afternoon and appreciate the privilege of sharing some of the work that's going on in the gene editing space, and specifically some of the progress that we're making at Intellia Therapeutics, which is a company based in Cambridge, Massachusetts. We've been in business now for 5 years and are very excited about the work that we do. What I'm going to try to do over the next few minutes is break the topic into 4 broad chapters. Just a word about the burden of genetic-based disease and try to give you a way to think about it. Talk a little bit about the elements of genome editing. It's a complex area. It's not going to be a molecular biological seminar, but some ways to think about the way that this work is done. Give you a sense of the progress that we're making at Intellia and in the field in general in terms of moving genome editing using CRISPR-Cas9 to the clinic, and then step back and talk about some points that I think bear some consideration as the field collectively moves forward. Now, we talk about rare disease, and I would point out that rare diseases collectively are not rare. They're prevalent when you add them all up. It's estimated that about 6% of the population here in Europe, and it's generally true, will be affected by a rare disease in people's lifetime. That translates into large numbers, about nearly 3 million here in the UK. And the preponderance of these illnesses are poorly treated or not treated at all. And they're expensive as a group. You see the National Health Services estimated bill to pay for these. And actually, much of that is spent just trying to reach a diagnosis as people tumble through the system without knowing, in fact, what the disorder is. When we talk about rare diseases, many of them are genetic in nature, and some of those are monogenic in nature. And it's been estimated that many thousands, in fact, as high as 10,000 discrete monogenic disorders might exist. Now, some of these have the same manifestation with different genes that present the same phenotype. Nonetheless, there's a lot of them. And so if we're going to be able to do something about this in a way that's generally applicable, it would be ideal to address the gene itself and find some way, in colloquial terms, to fix the broken gene. Now, here's another way of thinking about those numbers that I talked about. And what this is data that I drew from the OrphanNet database. And what it is the first 650 rare diseases arrayed by descending prevalence from left to right. Not all of these are monogenic yet, but there's a lot of these diseases, and I would point out there's an arrow to the right, where this is a very long list that continues. And I would call your attention to the prevalence, which is indicated in the y-axis, where the x-axis is 1 in 100,000 of the general population. As you click up from that, because this is a log scale, that next hash mark is 1 in 10,000, and above that 1 in 1,000 patients, and below the x-axis 1 in a million or 1 in 10 million. Another way of thinking about this is if you look to the far right and take one of those blue bars, for every patient there, where there might be a handful of patients in Europe, there's 10,000 to 100,000 patients in the blue bars on the far left. And so when you add this all up, this is tipped decidedly to the left. I mean, there are many, many patients on the left side, not so many patients on the far right side, but a very long list of diseases that continues far to the right. So it's estimated that in excess of 20 million patients will have in aggregate a disease with a prevalence in excess of 1 in 100,000, and a few 100,000 to the right of that vertical line. Now, we can pick out amongst these diseases the 100 most prevalent monogenic disorders, and I've indicated each of those with an orange line. And we won't go through all of them, but I put a few markers on there for some points of reference. You see retinitis pigmentosa, disease many people will be familiar with, cystic fibrosis, and I'll say a word or two about TTR amyloidosis, transthyretin amyloidosis, a disease that we're working on. And there in that first 100 monogenic diseases, hemophilia B, another area where we're working, does not make the cut in terms of 100 most prevalent diseases. And SCID, ADA, a very famous but very rare condition, is on the far right. Everybody's familiar with the bubble boy, but in fact there's a handful of these patients. So much of the disease burden is on a fairly short list of about 100 or so monogenic diseases. But if we're going to do something about the overall set of conditions, we have to realize that there are literally many thousands of them. So it would be ideal to have a system that's very adaptable, that's universal in some way, so we could think about addressing all of these. Now, I think that that's a very Mendelian way of thinking about the world. It's monogenic in nature, and that is a useful way to organize a lot of information, a way to think about these syndromes. But I think it's also limiting in some respects, because if we're able to think about modifying genes in general, we can do more than just fix broken genes. We should be able to alter normal genes so that cells can have their normal function changed so that it can do something that's desirable from a therapeutic point of view. So here's an extension of that sort of thinking. If we go beyond fixing broken genes and think about complex systems and how normal cells behave in them, we can start to think about other conditions that are also prevalent. So here is a list of the top 10 autoimmune diseases in the United States, starting with rheumatoid arthritis, where about 1% or so of the population has it. And you see in descending order there down to the top 10. Collectively, just this list has probably a third or so or more of all the patients that we just went through on those rare diseases. That's just 10 diseases. These are prevalent conditions, and they all result from a disordered response to some stimulus. The cells are normal. The genes are intact. We're not talking about fixing broken genes, but this is a complex system that we want to behave different. So if we could find some way of rewiring or redirecting cells so that they behave in a way that we want them to, we can start to think about changing what is otherwise an abnormal response. And similarly, that thinking applies to cancer, where, again, prevalent and frequently occurring conditions. What underlies the malignant condition is certainly the genetics of tumors, which are complex, usually not strictly Mendelian, but in the end, a failure of the immune system to respond to abnormal cells. The immune system doesn't recognize tumors, or for some reason those cells cannot respond to the tumor. But if there's some way of removing the impediment to that response genetically, we could think about modulating that immune response. And once again, having a tool that would allow us to rewire and redirect what are otherwise normal cells might enable us to think differently about using the immune system as a therapeutic. So when I think about gene editing, I tend to put it into two very broad categories. Gene editing is applied to diseases that are a result of a single genetic change, where the intention here is to fix the broken gene, and then diseases that are a result of altered function of otherwise normal cells, where the goal here is to somehow rewire or redirect how those cells function. So if we're going to address that genetically, if we're going to intervene at the level of the gene, we need a particular set of tools and a set of very specific conditions. And although this is an oversimplification, I do think it's a helpful way to think about circumstances in which gene editing might start to be applied. And these conditions, I think, generally speaking, need to coexist if we're going to be in a position to apply this sort of approach. First and foremost, we need a tractable disorder. And by that I mean a condition that's chronic and progressive in nature or that's reversible. I'm not talking about an embryological problem where there's disordered anatomy or histology that cannot be reversed. It needs to be something that we can actually do something about. Secondly, we need to know the nature of the genetic problem. We need to know the causative gene, how it works, why it works, and what to do about it. And if we're going to change a complex system, we have to have some sense of what that protein or that regulatory element does as a linchpin in that overall systemic response. Third, it needs to have a target tissue. And think about it. This ideally would be a tissue in which the causative gene is expressed, and it's limited to that, and we can actually access it, as opposed to a gene that's disordered in every single tissue of the body, which makes it very, very difficult to reach and affect some sort of therapeutic outcome. And last, and probably most important, is having an editing tool. That is, some means by which we can deliver to these cells an ability to affect the change that we're looking for. And up until now, up until the last few years, I know we've had some pre-existing technologies, which is entirely fair, but they have not caught fire and they've not been widely applied. That editing tool has been largely absent. So all four elements, in my estimation, really need to coexist if we're going to hope to use gene editing. Now, Jennifer Doudna, one of the scientific founders of our company, famous for her work in CRISPR-Cas9, has made this statement, which many coworkers and certainly people in the academic environment agree with. And that is, we believe that we may be nearing the beginning of the end of genetic diseases. So what is driving that enthusiasm? What's primarily access to this gene editing tool and the utility that it brings. And the origin of that comes from what was referred to earlier, clues that exist in nature that we've now better understand and are able to deploy. And I'll just give you a very quick primer on where this comes from and how it's used. And it begins this way. CRISPR-Cas9 is a system that exists normally in nature, and it comes from bacterial systems. And the life of a bacterium, very simply put, is represented here. Single cells living in an environment that are barraged by bacteriophage on a daily basis. When a bacterial cell is infected with a bacteriophage, we all know that the cellular machinery is taken over by that virus. Rounds of reproduction take place, progeny virus is produced, and it's very bad news for the bacterium because it lyses and dies as those progeny virions believe to infect subsequent rounds. But there are circumstances where this can be avoided, and that's where CRISPR-Cas9 comes in. So CRISPR, which I've been referring to here, is clustered, regularly interspaced, short palindromic repeats, a mouthful, which is why everybody says CRISPR. And imagine a different circumstance where we now have that same bacterial cell, except there's a couple of differences. And the first difference is that lying encoded in its genome are short sequences that exactly correspond to DNA sequences that are present in those infecting viruses. And also, there's a gene that encodes an enzyme, a nuclease called Cas, which cuts DNA when it's directed to a particular place by an RNA copy of one of those DNA sequences. So if you look at the bottom on the far left, there's the nuclease, cuts DNA, and it's waiting to be armed by that RNA copy that is an exact sequence copy of the infecting virus. And together, they form that green blob up there, which is the guide protein complex, which is able to now cut DNA. So back to our bug in the first place, except now it has this adaptive immune response. As the viral particle infects a cell, now because of the preexistence of this guide protein complex, that DNA within the virus is recognized, it's cut, it's disarmed, it's inactivated, the virus is not able to reproduce, and the cell survives. That is the essence of what is CRISPR-Cas9. And what's so exciting is that we can co-opt it and use it as that editing tool for the work that we want to do that is outside these bacterial systems. And basically it boils down to this. It's a two-part modular system where that Cas9 enzyme is generally applicable, it can be held constant, and it can be armed with a guide directed against the sequence of our choosing. It doesn't have to be against that bacteriophage in the first place, but against literally any stretch of DNA. So that when those two elements are put together, you now form essentially a scissors that cuts DNA across the double strand and does it in a very, very specific fashion as a result of that 20 nucleotide sequence in that guide RNA. And what's particularly exciting about it is in those brackets, in that box, is that there's literally an infinite number of guides that we can ultimately choose to direct this activity against virtually any gene of our choosing. And that's humans or any source of DNA. So that tool that we've been looking for, that's potent, selective, simple, universal with respect to what it can go after, it can introduce one or multiple edits and do it on a permanent basis, is now in hand, and it's CRISPR-Cas9. So let's think of this in terms of the types of edits that one would want to introduce. And there's a lot of molecular biology that one can go through. But I think of this in terms of what I would call archetypal edits, the general categories of things that one might want to accomplish. So you've got at the top the Cas9 enzyme, that nuclease, armed with an RNA guide. And there's three sorts of things that one might generally accomplish. One is a knockout of a gene where you introduce a double-strand break. As a cell rushes to put that together, it's knocked out of frame, you introduce a stop codon, and the gene is disabled. Or you can actually excise parts of that gene and remove it entirely. Second sort of edit would be a repair, and this would be where a short sequence of DNA would be introduced to repair a codon or two. And then third would be an insertion where, once again, where the double-strand break is, one can introduce DNA up to kilobases and essentially reconstitute a gene or genes in a very specific place. So three types: knockout, repair, and insertion. So back to where we began, which is thinking about fixing broken genes or rewiring and redirecting cells, which is where the field is going as we think about how to deploy this technology. And I'll show some of the differences because I think sometimes people confuse these different sides. It all starts with CRISPR-Cas9, and it's a function of how you're going to deploy it. You can deploy it either in vivo, where CRISPR itself is the drug and is the therapeutic agent. That is typically for genetic diseases, those Mendelian disorders that we talked about at the beginning. And Imagine an intravenous infusion where you have a formulated form of the RNA coding sequence of the Cas9 enzyme with that RNA targeting guide. That is the drug. That is what we do. Or you can go the other side where instead of taking CRISPR into the body, you're taking target cells out and modifying those cells in a dish. And it may be multiply edited, singly edited, whatever. But the CRISPR is left behind, and the now altered cells, which were normal in the first place, but are now, I'll say, rewired, are then infused back into the patient. And those would be used typically for immuno-oncological or autoimmune sorts of disorders. So 2 different approaches, in vivo, ex vivo, CRISPR is the therapy, or CRISPR creates the therapy. Well, let's talk about some of the progress that we're making and a little bit about that paradigm in terms of where we are as a company and generally speaking where the field is, and talk about some examples of where we attempt to fix the broken gene, again, by going in vivo into patients. Well, back to this notion of a set of conditions having to be present. It turns out that the liver is a good example where you can find all four of these elements. There's a series of tractable disorders. Remember, these are chronically progressive or reversible. We know the causative genes of many of these disorders. We have conditions that are isolated to genes that are expressed in the liver, and we can reach the liver from a delivery point of view. And finally, with the editing tool in hand, and knowing something about the structure of these genes, we know that these can succumb to these archetypal edits that we are talking about. And so I've chosen a list of some of the sorts of disorders that people think about and some of which we work on. And to give you some examples, transthyretin amyloidosis, which we'll talk about in a moment here, has TTR as its target. This would be a knockout. Can you disable the gene? The hemophilias, A and B, we know what the genes are. This would be a question of can we reconstitute the gene to make the missing protein? Alpha-1 antitrypsin deficiency, there's a single codon that creates much of the problem for people with this form of inherited COPD. This would be a repair, and so on. So we can find a set of conditions and disease states in the liver that all come together in a way that the gene editing approach for monogenic disorders on an in vivo basis should be addressable. So let's talk about one, and this is one that we're actively working on in Intellia, and this is transthyretin amyloidosis. We call it TTR for short, which is treatable with a gene knockout in the liver. You see the incidence there is about, or actually its prevalence is about 50,000 patients worldwide. It's a mortal illness depending on when you're diagnosed. You see life expectancy there. It's a disease that's caused by the accumulation of a protein, typically in the heart or nerves. Cardiomyopathy results, or neuropathy, which is lethal in the end. Dominant condition, and even though there's over 100 discrete mutations within the TTR gene, disabling that one gene should be in a position to address all of those underlying mutations. And of course, the gene is expressed in hepatocytes. So the archetypal edit that applies in this case is a knockout. So we stop the production of that pathogenic protein, and this formulated form of RNA that encodes Cas9 with the guide RNA would be the way to address that. So a little more detail, but it's in essence what I just said. Imagine the affected TTR gene at the top. It produces a protein that doesn't fold right. It's deposited in heart and nerves. So the goal here is to disable that gene, introduce a double-strand break, have it come back together in a way that has a stop codon, then eliminates production of the TTR protein, and with that, we treat the patient. So we're not in the clinic yet. We expect to be doing that next year, but here is very advanced preclinical work that shows exactly that. So this is work done in non-human primates, monkeys, in which a single intravenous infusion of those LNPs targeting TTR is given to a set of animals. So there's control animals on the far left, and I'm looking at that far left panel, which measures editing, and treated animals right next to it. And what we're doing is measuring the proportion of alleles that encode TTR that are altered or edited in the liver. And you see that, again, after a single infusion, about 75% of all of those alleles are edited. They have an indel that's been introduced, which renders the gene ineffective. Now that corresponds to almost all the hepatocytes. There's other cell types present in the liver besides hepatocytes, but the preponderance of these cells are in fact hepatocytes. And when we go to look for presence of the protein that results from this gene, which is illustrated on the right, we see that control animals for which editing did not take place continue to produce their TTR, and animals that have received the treatment have now had their TTR gene edited. You see the plummet of their TTR circulating levels in those blue lines. That's about a 95% or so reduction from baseline. This is 6 months of observation. The observation continues. We've got animals out to 10 months. We know that we get to precursor cells. The expectation is that this will be a permanent change. And just as a point of reference, that blue box up there is generally regarded as therapeutically relevant. If you can reduce TTR levels to that range, and we are well within it. So imagine a circumstance here where potentially in humans, if this can be reproduced there, that a single dose, a single administration would alter this gene in a way that this pathogenic protein is no longer produced.
John, if there is a pointer there, if you want to use it on the...
It was the, this was the, oh yeah, okay, thank you. Now I've got all the equipment. All right, good, thanks. Another example is hemophilia, which I said earlier would be treated with a gene insertion, not a knockout. We know that this is an X-linked disorder. You see the incidence. It affects boys, men, primarily. And factor IX is the missing protein. You know, in particularly severe cases, this results in even spontaneous bleeding, and it's treated by replacing factor IX. So if we could introduce the gene that produced the normal factor IX protein, presumably these patients would not have to receive that factor IX therapy and might even potentially be cured. So genetically, what we're trying to do is insert somewhere in the liver a gene that will produce that protein. And now the edit is a little bit more complex because we need to not only cut the DNA, which we do with our CRISPR-Cas9, does only cutting, but we also need to supply a template of the normal factor IX, which we do in this case virally with an AAV factor IX DNA template. So cut and then introduce the DNA. So a little bit more about the genetic architecture just to make that come alive. Here is an example of the coding, the early portion of the albumin locus in the liver, in hepatocytes. What you're seeing are coding sequence in the various exons and a non-coding sequence here. And we're able to target these LNPs using CRISPR against a particular section of the non-coding region that we've selected for all kinds of very particular reasons, and use what is a very, very potent promoter. Albumin is one of the most expressed genes that there is in the body, to drive whatever DNA insert we provide. And here you see that insert, which generically is represented as a DNA template, which is bidirectional, so that independent of the orientation, left to right or right to left, you should reconstitute a normal gene and be able to have it in frame. So after these two agents are delivered simultaneously, we cut in the albumin locus and introduce this factor IX coding region, and with that are in a position to produce factor IX protein. So how do we do? We take this and apply it preclinically. Again, these are non-human primates. We're not yet at the clinical stage. You see a series of controls here where, as expected, no factor IX is produced. But when both the LNP and the DNA template are provided simultaneously, within 2 weeks, we're producing what are normal human levels of circulating factor IX protein. And again, one would expect that this will be a very durable effect. We're following these animals and continue to do so. And in contrast with typical gene therapy, where a virus is used that provides a gene but does not integrate into the chromosome, can be lost over time, this would be expected to be a permanent effect and therefore potentially applicable to young children who need to grow and have their hemophilia controlled over the duration of their life. So let's move to the other category, where conditions are diseases that are a result of the altered function of normal cells. We're not fixing broken genes now. This is this idea of rewiring and redirecting cells, changing how cells function. I'll give one example. Again, the principles are the same. The nature of the edit, the archetypal sorts of edits that I've described are applied, except now to change the DNA to introduce some new functionality that was not there presently before the intervention. So the case here would be AML, which you'll know is a common leukemia in adults with a terrible prognosis. See the incidence of this in the UK, about 3,000 or so patients. And simply put, despite the complex genetics of the tumor itself, in the end, I think the disease can be portrayed as a failure of the immune system to recognize those abnormal cancerous cells. And that's in part because lymphocytes are really unable to see and react to those tumor blasts. And that's largely because the T cell receptor that is there is blind to epitopes that are presented on the presence of, on the surface of those tumor cells. So if we could somehow change the T cell receptor that was there in the first place with one that's able to recognize peptides present on the surface of these tumor cells, we would be in a position to change how these cells react to that tumor and respond. So the task here is to introduce a new T cell receptor that in this case recognizes a particular epitope of WT1. Now it gets more complicated, but again, the same sorts of changes are being made. We're using the same sorts of editing techniques. And essentially what we're trying to do is remove the endogenous T cell receptor that was there in the first place, and in place of it, introduce a new T cell receptor of our choosing. And there's a science behind this, where that TCR comes from and how we select it. Let's leave that aside. The real question is, can we do this? Can we get it in there? And so the basic idea is that because the T cell receptor has two chains, alpha and beta, the first order of business is to knock out that first chain. And we do that by introducing the construct that we've made that encodes both a new alpha and beta chain for the TCR of our choosing. So we can knock out one chain, introduce the coding sequence of what we want in one fell swoop, and then secondly, part two, knock out the beta chain so that the endogenous T cell receptor is gone, our new coding sequence is put in place. So how do we do? Lots of data that we could go through, et cetera, but it's essentially summarized this way. What you're looking at are the results of removing the endogenous T cell receptor here on the left. So the alpha chain, the beta chain, or both chains together are essentially completely removed from these cells, which is important to avoid graft versus host disease or to have a mispairing of these chains with whatever TCR that we might want to introduce. And then secondly, after we introduce the new T cell receptor, the question is, does it work? Does it kill blasts? That is, the ultimate target here. What you're seeing here are a series of experiments where unmodified versus these introduced TCRs show high levels of killing against AML blasts. That's very exciting because if that also plays out in vivo in patients, that potentially is a very powerful and effective tool to deal with a malignancy that's poorly treated right now, despite a lot of work that's underway in a variety of different settings. So stepping back, when we think about altering cells in terms of rewiring them, I like to put this in four kind of canonical ways of thinking. And this is with respect to lymphocytes. Just heard a little bit about T cell receptors. There's any number of T cell receptors that one should be able to introduce. It's a matter of choosing them and whatever the circumstances are that one wants to deal with. One can also pursue ways of making foreign cells or donor cells close to universal, so that instead of having an autologous source of T cells, one can look to other donors as a supply of these cells. And you can do that by very systematically looking at that array of different signals that drive recognition of foreignness, which we are doing, so that ultimately you'd be in a position to combine your T cell receptor platform with an allogeneic chassis, if you will, and have any array of different lymphocytes that have been modified that could ultimately be produced. By the way, I would point out that despite the enthusiasm we have for CAR T cells, which are related but different, putting an antibody on the surface, and that has numerous limitations, cracking this is what will ultimately drive the cost down. Manufacturing circumstances will radically change, applicability will vastly expand, treatment intervals will decrease between the time of diagnosis and administration, and I think this is a very, very important way of ultimately improving patient outcomes. And then there's other forms of research that we're actively doing where we look to modulate the degree of activation of lymphocytes and look for those cells that determine the extent to which lymphocytes enter solid tumors. All of these different edits, all of these different approaches can ultimately be combined into single cells. Early work is here. This is rapidly coming. This is a little further down the road. It's all on the way. So stepping back from this therapeutic side, as we are standing on the verge of this work going into the clinic within the next year or so, I think we can conclude the following. We now know that we at our company and our compatriot companies are all able to edit cells in a very selective way, and in some circumstances, with work I've shown you, in selected tissues in vivo. We've done that in animals, and in some cases now we actually have CRISPR-based therapies that are entering the clinic with the other companies. I've shown you that we can activate disease-causing genes in the liver, we can introduce corrective genes and express them in the liver, and we're starting to deconvolute ways to modify complex cell systems, with the immune system being the first order of business, but you can imagine this generally applied across any number of different approaches. And as I look ahead, what I see is the following. I think, and in fact I know, that there are multiple programs that will be in the clinic in a matter of months to a year or so. A couple are already there. And I would expect that we will start to see some of the first human data, perhaps even by the end of this year. That's not work done by Intellia, but it's work done in hemoglobinopathies, and it should be coming in short order. You will see an expansion of work in those addressable tissues. I talked about lymphocytes, cells we can take outside the body, talked about the liver, which is addressable. There's a lot of work going on to find ways to get to other tissues that are a little harder to access at this point in time. And finally, it's not three companies just doing the work. There's a vast academic enterprise that is accelerating this and feeding it and bringing their own expertise. And I would expect to see a flow of information from those organizations as we move forward. And so I fully expect that there will be CRISPR-Cas9 edited products available for patients, for doctors to write prescriptions in just the very next few years. So stepping further back, that's all happening. That is coming. It is real. It is on our doorstep, so to speak. I think there's a few broader categories that bear comment, at least. One is just thinking about medical uses and what's appropriate, going back to this notion of very rare conditions. I think we have to acknowledge germline treatment and what that might entail. We should talk a little bit about non-medical uses, but nonetheless in humans. And then finally, I'll say a word or so about non-human uses. So what do I think is an appropriate use of the therapy? I do not see gene editing displacing effective, inexpensive, drugs that are reversible for which, we have good outcomes. I think that would be crazy. It would be a misallocation of of resources, and the risk-benefit profile would definitely not warrant that. And so, if you're taking antihypertensives, you're going to keep on taking them. We're not going to solve that problem in the near future. But you will see ongoing work continuing for what have been traditionally undruggable severe genetic disorders. The autoimmune disorders, as I've already talked a little bit about, advanced cancers. And then I think you're going to see people talking about how to address risk factors. And I think that's a very interesting sort of area where risk and benefit really come into play. We have to think very, very carefully about that, because think of it this way. Even though we may know the gene that gives somebody risk for some disease, if we don't know that a person who carries that gene will actually get that disease, we'll find ourselves in a position where we need to treat more people than will actually derive benefit. And so we could find ourselves in a position where we expose lots of people to risk, but very few people to benefit. And I don't think that's the right use of genome editing until we get a lot more information about how these therapies behave and how these patients perform over a long period of time. And back to this notion of very rare diseases. Part of the appeal of this approach is that in the end, it is the ultimate personal therapy. I mean, we're literally talking about individual gene sequences with potentially you could deliver a therapy to one person who has an individualized therapy for whatever it is that we're attempting to treat in that patient. This would be, in the ideal case. And so when we think about traditional approaches to drug development, clinical trials, the economic environment in which all of this exists, I think there's some circumstances where the traditional system that we have actually works, and that's where all the companies are beginning. But as we tail off into these disorders that are increasingly smaller and smaller patient cells, even though we may have a way of approaching them that's in hand, and these have traditionally been forgotten people, it may be that either the clinical trial apparatus that we use or the economic incentives that drive this prevent this from happening. So I believe that if we can find ways to extrapolate from work that we do on this patient population and somehow build it into these increasingly smaller groups of patients, we may be able to stretch a little further to the right. But that requires a lot of work together with companies, regulators, scientists, et cetera. And it's interesting when you think about this. If there's a patient population of 20 people, let's say, the test may be the treatment. I mean, literally, as we expose people to a therapy, if it is truly curative, once we've exposed them to the therapy, it's done. So from a traditional sort of economic incentive, the market disappears. So that's another aspect that I think needs to be carefully thought through. I want to say a word about germline treatment because it's a topic that comes up frequently. I'm frequently in positions where either it's a lay audience or the media will have a series of questions that immediately go to this particular topic, which I think misses the primary use and 99% or more of all the activity that's going on in this space, which is to treat somatic disease. Every company that I know of is limited their work to this. There's many patients there. There's lots to do. But just to make sure that we're all talking about the same thing, in the case of germline therapy, we're talking about treating, usually via IVF, some developing embryo, so that a patient is not being treated, but some future generation is. And that is an undertaking that there have been some very spectacular cases in the media that we've heard about, but this is not the work that's taking place in the industry or most academic environments that I'm aware of. I would also point out that from the standpoint of germline treatment, it's something that in most circumstances is actually not necessary. There are some examples where it absolutely will be, But when you think about most Mendelian disorders and ways in which we would deploy it, it begins with an act of in vitro fertilization and then some pre-implantation diagnosis. And at that point, you have a choice. You know diseased embryos or those that are not diseased. And you can choose a diseased embryo and edit it, and discard the normal cells, or you can choose normal cells, implant that, and discard the affected cells. This side, I think from the standpoint of benefit risk, seems like an obvious choice, but of course it won't apply for every single circumstance. So as we think about germline editing, I think it's important to think about what the process actually is and what it, the nature of the problem that we're ultimately trying to solve. And so there are circumstances, but not so many when you come at it from a Mendelian point of view. But we believe that research should definitely move forward. And we need to understand how to use this and to do it in a way that is responsible and effective. Now, so we think about non-medical uses in people. This is typically put in terms of enhancement, okay? And this is, I like The Economist, I read it a lot, and this particular cover from a couple years ago caught my eye. Nowhere on there does it say cure cystic fibrosis or, sickle cell disease. It's all about improving IQ, color of eyes, and so on. And I think that typically in these cases, not always necessary, but typically we're talking about germline modification here, or at least a developing embryo. And we need to bear in mind for these discussions the technical issues where this is very early stage research and there's a lot of things that we don't understand. What is benefit risk in circumstances like this, I think needs to be thought about very, very carefully. And there's an assumption that these genes act in isolation, particularly as an embryo develops. And in many cases, we don't really fully understand that. Secondly, or I guess not secondly, but down the list here is what is consent and for what purpose? When we talk about improvement, in whose eyes? So I don't have the answers to these questions, but as the technology improves, increasingly these will be points that we talk about. And then finally, stepping even further back, non-human use. The approach is universal. We can edit DNA in whatever form we find it. And I think we will hear, and we already have in many circumstances, that there's all kinds of things we might do for human purposes, in fact, for human health, whether it's to improve the food supply, make more protein, or have animals behave differently so you can put more of them in a smaller place, typically, or even find the source of human pathogens that require us to alter the environment because it serves some human interest. These are very complex relationships and have very complex issues. They're easy to do, technically, in many respects. And so I think this is an area that we need to stay very actively informed on and engaged with. So, in summary, I would say the following. This is very powerful stuff, and it's happening fast. It is, as a physician, very exciting because we can expand the pharmacopoeia in a way that was unimaginable just some years ago. We've only begun to explore the capabilities. These are the early days, and as we were talking at lunch, imagining what the world is like 20 years from now is just, I think, mind-boggling. But it will test the limits of some of our standard assumptions. Benefit risk must always be borne first in mind because these things cause permanent changes, and there's much good that we can do, but there's many things that we may not fully understand. And like it or not, it's here to stay. And there's going to be a lot of it in our future. So there we go. Thank you for your attention. I appreciate it.
John, that was a terrific run around a very exciting field and a very comprehensive coverage. So we've got some time now for discussion about the issues that John's raised, and there are two roving microphones. So raise your hand and a microphone will reach you. Could I kick off with the haemophilia application that you referred to? Two questions around that. One is that if you, as you're suggesting, use the, or have you, as you have done in experimental animals, use the albumin promoter, how are you going to control the amount of haemophilia that's produced? But the immunologist's question is, you're introducing now what is essentially a new protein into an adult, or at least an established immune system that has no tolerance to it, indeed might be sensitized to it because you might have been administering factor 9 or factor 8 to these patients. So how are you going to manage a potential autoimmune response against a neoantigen?
Yeah, so, we chose the albumin as promoter as the first case, which doesn't mean it's the last case. And I think the ideal ultimate use would be to exactly, as we did with the TCR, put it in locus so that it's driven by its normal sorts of physiologic processes. And we were very gratified with the high levels of protein that were produced, and that's without optimizing it. You can influence how many cells you ultimately edit. This is probably a minority of them. We're still determining the exact number. We've shown in mice just a few % will produce lots of protein. What is your dose of virus and so on. So there's clearly work to be done, but I think we believe that there's a lot of predictability with respect to how these things behave, which we've learned through the other systems. Now, with respect to immunogenicity, we will see. And certainly the gene therapy people are leading the way here where, they're introducing foreign proteins as well, using a virus to do it. It's not integrated. I think in many cases they've been quite successful. It's a tolerance. You're more of an immunologist than I am, but certainly you can get a degree of tolerance. And in the liver in particular, I think it's some unique attributes there. So we will see, but the early results are certainly encouraging. And in monkey, which clearly this is a foreign protein, there's been no response that way.
There's lots of questions, so just...
John, you made a good point about the prevalence of these diseases when taken together, but of course individual efforts have to focus on single diseases. And in the monogenic space with conventional gene therapy that you touched on briefly, I wonder what your views are on the commercial model, because these diseases are so rare. And we've seen the approval of the first few of these monogenic gene therapies. But these diseases are very rare, and I've been involved in a couple of them, and I just wonder what the commercial model, with an industry perspective, and obviously talking to colleagues whose job is to try and commercialize these products, how is it going to work?
Yeah, I don't think the current model that we have is going to successfully treat that tail, far into the tail. And it becomes a question, I think, for all of us, so it's what are the, I'll say, shared responsibilities about doing something about that, because that's a lot of people and it's a lot of cost. There is a social good to be gotten. And I think we hopefully can come up with some models that are a little different from the ones that we typically have. We have some ideas. I briefly referenced it in terms of, is it possible to decrease the amount of preclinical work that's done? Because every one of these, we think about from a regulatory point of view, which is the system that we use, it's appropriate. I don't mean to be dismissive of that in any respect, but the degree to which we can extrapolate on work from prior examples, I think is an interesting thing to think through. So, case in point. That LNP that I talked about, it's the same LNP, for any hepatocyte. And so how many different diseases are in hepatocytes? Well, we could tally that up. The coding sequence for the nuclease is the same coding sequence. And literally, the only thing that's being changed is a 20 nucleotide sequence. So are there some efficiencies we could get there? And then what is the cost? What is enough to drive the incentive? And, we can't we can't charge $5 million and $10 million for every one of these patients, I don't think. It's just not going to work that way. But if we find some degree of predictability in enough of these patients where we can imagine going from one to another, which I think is the challenge here, I don't know if we're going to solve the very end of the tail, but I think we can move further down it. So,
Nilesh.
Thank you. So targeting liver expressed genes is obvious, but what about other tissues? I particularly mind, inherited cardiac diseases. Do you see that? Can you tell us your views of whether this is ever going to get there?
well, I want to get there, and it's go back to my 4 circles. This is, there's the edit, and then there's the delivery, right? And those are 2 fundamental issues. And delivery is an area of intense interest, right? So the liver, in a way, is cheating a little bit, because if you take a lipid nanoparticle, the liver loves lipids, and it very actively takes it up. But can you use that as a point of departure for engineering in minor changes to identify receptors on these other cell types? We're doing that work so that ideally you can get to muscle cells for Duchenne's, for example. There's a lot of work going on there. Or can you get to stem cells in the lung for cystic fibrosis? We're not active in that space. But these are the sorts of questions, and it's in some respects, it's verges on sort of a traditional kind of medicinal chemistry problem, which is, can you generate molecules that have specificity for some particular protein that may delineate a particular cell type? So, it's early days. I'll go back to my 20-year horizon. I will be astonished if 20 years from now we're working on if we're not working on a whole host of tissues outside the liver.
Yeah, and then we'll go over the sides, so don't worry, I'm not ignoring you.
Jane Spink, Genetic Alliance UK. You made a really clear distinction between somatic and germline and explained the reasons why germline is unnecessary in the context of PGD and embryo selection. And I just wondered, on your 20-year horizon, whether there might be scenarios you can imagine where in utero therapies in concert with prenatal testing might be advantageous, and where you felt the risk-benefit balance would sit with prenatal.
Yeah, I hope so. I do, and it's there's technology to be developed. With respect to the germline, I'm not trying to ignore it in any way. I just think it's not ready for prime time. And I think there's circumstances where people talk about using it where it's just not necessary. For conditions that we can recognize in a developing fetus, for example, and if we could intervene in a way that's, I think we should absolutely try to find ways to do that. And that's not an active area of research for us, but I think that's one that should definitely be pursued in the academic environment, because those are very important technologies that could prevent disease once we first recognize it. So in 20 years, when I think about 20 years of my lifetime, what I've seen from a technical point of view, yeah, I suspect that there'll be real progress there.
If I could just insert the fact that there's a consortium formed between the National Academy of Medicine in the US, the Royal Society, the Academy of Medical Sciences here, and the Chinese Academy, I think it is, looking at what would we need to know before we considered germline editing. So that'll be an important piece of work. Yeah.
Paul Williamson, University of Melbourne. You commented about the availability of pre-implantation genetic diagnosis, which we've been offering now collectively for 20 years perhaps, for cystic fibrosis, thalassemia, and so on. When this is done, as you know, for medical reasons, you often finish up with 6, 8, 10 embryos that are unaffected by the genetic disease, and patients are already, families are already beginning to ask, can we do tests and can we actually intervene in some way or another with these embryos? It's not a question of developing the technology in order to apply CRISPR. The technology is there and being used. Would you have governments intervene and prevent the application of CRISPR technology in this context, where people have 6, 8, 10 embryos and actually want to do something in that context? And when would you, if you would prevent it, why? And when would you stop preventing it?
Could somebody write those down for me? I'll take one sort of shot at that. I recognize the technology exists, and that's what I was saying. Because it exists, we should think a certain way and use it. And I think that's entirely appropriate to make decisions to discard one or the other type. And I think that's the point I was trying to make. I don't know what the regulatory environment is in the UK or in many European countries.
14 days, I think we can keep an embryo going.
Yeah.
To experiment.
Okay, but I was going to think about just even, yeah.
I think, was that your question, Bob, as to what the regulation should be of research on embryos using this technology experimentally?
I think that there's a tendency to fail to realize that this is a situation that is already very, very close because of the way in which it interacts with existing technologies. And I am asking whether you feel that the regulation at the moment should say that this should not be done, and if so, why and
That this being editing
this being
based on, yeah.
editing of embryos?
Well, it's the reasons I gave. I think that in most circumstances, it's not necessary. And if this is a very, I think, immature area where it's easy to assume that we know what the outcomes are, when in fact we've barely begun experimentation. So when it comes to thinking about it in terms of benefit risk, which is how I think everything, medicine should be considered, let's make sure we have some understanding done in a systematic way of what the risk is. And I'm not saying don't do research. I think there should be a robust research program. I'm all for that. This is not about darkness. This is about light. And but I think the temptation to act on what we want to be true based on what we know is strong in some circumstances, particularly when you just look at the setup, which you just properly emphasized. It's not necessary.
Lizzie. Oh, Victor, okay.
Does the CRISPR system make mistakes, or is it 100% accurate?
The only thing that I know that's 100% is physics, I suppose, gravity, in death, taxes?
Death.
Okay,
Death,
that's okay. So no, there's nothing in biology, nothing thermodynamic that is 100%. But if you're asking, are there off-target effects? The answer is yes, but you characterize them. And so part of the exercise here is to find those particular guides that have the performance characteristics that we want. If one wants what we call dirty guides that there may be a redundant sequence, or across those 20 nucleotides, 19 of them match, or 18, that kind of wobble sort of thing, there are ways that have been worked out. This is a regulated thing. There's consortia that address this to assess that and to determine the degree to which it's there. And then in the end, it comes back to benefit risk. So our job And we take this very seriously, is to find clean guides that behave the way we want them to. That's what the company does.
Lizzie.
There are about 6 million people in the world with trisomy 21 Down syndrome. We've had prenatal diagnosis for a long time, with the lovely paradoxical result that in some populations the incidence of Downs is actually going up as people have the prenatal diagnosis but feel relaxed about having a child with Downs. So I wondered, very, very unfairly, if you could speculate wildly about this sort of treatment for the chromosomal disorders.
I think I'm off the hook with all those disclaimers. I don't know how to do it. I really don't. You know, and that's a very sad thing, which is why when I was giving those sort of 4 categories of things, I said a reversible disorder, a target tissue where you have, you know, something that's, you know, disease that, or a gene that's expressed in a particular tissue. I think we're very challenged with respect to the sorts of conditions that you're talking about. And I don't know how, based on what is the nature of that, you know, particular condition, how CRISPR can help. And I wish I could say otherwise, but, you know, there's more work to be done, and probably, I hope, other modalities that may ultimately address that.
Yeah. Maybe this is the last question.
You just came back to the reversibility, and many of the monogenetic diseases probably give you a chance to stave the disease, but probably not reverse it. How much of a consideration is that when you triage the diseases that you choose to go after?
Yeah. There was a physician who joined our company who's an endocrinologist. And on his first day of work, I printed out a list of all the inborn errors of metabolism. I think there were 800 or something like that on the list that were, you know, tied to the liver. And I asked him to triage them along the lines of what you just said. And his first pass was there were fewer than 50 that kind of hit my criteria, right? Now, my job is to see if there's ways to extend that list, right? But, you know, the conditions I said are key, you know. I mean, even preventing progression, and I would cite the amyloidosis case, is potentially life-saving. You know, it's different, it's not curative, it doesn't take you back in time. But when patients are offered that choice, I think I know what they'll choose.
Great. Well, John, thank you so much. That was a really wonderful run around. Could we give another thanks to... Okay, so we're doing pretty well for time, and we now move on to 4 short talks. I trust they're going to be short anyhow. And I'm going to introduce all 4, and then if you come up one after the other without me having to come up and take the microphone again. So first up is Dr. Matthew Garnett, who's a group leader in the Wellcome Sanger Institute. He'll be discussing the role of genome editing, understanding drivers of diseases like cancer. Second up will be Nicola McCarthy, who's a business unit manager in Horizon Discovery, looking at next generation genome editing research tools. Thirdly, we'll have the patient voice from Dr. Alison Kay, the impact for patients and families that treatments of rare diseases would bring. And finally, Dr. Sarah Chan, Chancellor's Fellow from Edinburgh, who'll be discussing some of the ethical considerations. So over to you 4, and then we'll have another discussion at the end of the 4 talks. Sorry? Yeah.
Good afternoon. So what I'll be doing, I don't have any slides, is probably just touching on and emphasizing some of the points that John has so nicely raised in his talk. So just to really start out, you know, genome editing, and I think more specifically the use of CRISPR for genome editing, is a really disruptive technology at the moment that is empowering many avenues of biomedical research. And so I'm an academic scientist, and so I really wanted to focus my few minutes here on the application of genome editing for the study of both rare and genetic diseases, including cancer. And cancer is my own area of expertise. So the ability to change the code of DNA, and therefore the instructions of all life, is a truly powerful technology. And in the laboratory, we can use genome editing in multiple different ways to modify the DNA of cells. And as John highlighted, we can do amazing things. We can insert or remove different components of DNA. And we can make very precise individual changes in the DNA sequence. And we can actually even move large chunks of DNA around the genome of an individual cell. And not only this, we can actually control the expression of genes without actually even changing the sequence of the DNA itself at all. And all of this can be done much more rapidly, much more efficiently, and at a scale that a few years ago would have been absolutely unimaginable. And this toolkit is rapidly growing. There is a whole community that are passionate about developing tools. And with that, so are the applications. And there are many varied and creative, and I want to emphasize this, you can do so much with this tool that scientists are using genome editing to probe human biology. And I think what also makes it so transformational is it's very robust. It works really well. It's pretty inexpensive, at least compared to many other things that we do in science. And it requires a relatively modest infrastructure, which makes it accessible to many, if not most, laboratories. And so as a consequence of all these factors, it's been a technology that's very widely and rapidly being adopted by the research community. So scientists are using genome editing to tackle some of the biggest challenges we face in our understanding of genetic diseases. And so I thought I would just illustrate this with two examples of how we're using it in the kind of preclinical research setting. So we now have a wealth of information about the genetic basis of most or many human diseases. But the problem is our genomes are vast. So each of us and each cell, our genomes contain over 3 billion bases or base pairs. And the genetic sequence of every person is different in thousands, if not millions, of very small ways. What this means is actually distinguishing which genetic changes are involved in diseases as opposed to those that are naturally occurring within populations of people is really, really tough. And even when we know which genetic changes occur in diseases, determining how and why and what they're doing is actually extremely difficult. And so genome editing is helping us to solve this puzzle by allowing scientists to introduce these disease-associated genetic changes into the DNA of cells and study the effect of those changes on proteins, cells, tissues, and even at the level of organisms. And we can do this much more rapidly than we ever could in the past. And so this is happening for diseases such as inflammatory bowel disease, cancer, and Parkinson's, and to understand things like developmental disorders in children. I'll draw on my second example from my own work, which involves cancer drug target discovery. So using CRISPR gene editing and cancer cells grown in this laboratory, we have knocked out individually every single gene in the human genome, all 20,000 genes, and measured the impact on cancer cell survival. And we've repeated this process across hundreds of different cancers. This has revealed many new drug targets that kill cancer cells, including a protein named Werner helicase. And consequently, of this type of work, there is now considerable interest from the pharmaceutical companies to develop drugs that target Werner helicase for the treatment of some types of cancers. And finally, I'd just like to emphasize some of the distinctions that have been made here today already about somatic cell genome editing versus germline genome editing. This is important because the safety, the ethical, and the societal issues associated with each are quite different, and as are the rules that govern them. So just to be clear, somatic cell gene editing is done in cells that are not involved in reproduction. And therefore, the risks associated with this are lower, although I should say they are not negligible. And in fact, as was already commented, today this represents the major use of genome editing, both for basic research as well as translational applications. Whereas the germline editing involves altering cells involved in reproduction, such as the egg, and so these effects are passed on for future generations. And so research into germline editing is important, particularly if we do want to develop therapies to treat these heritable genetic diseases, where we want to alter all the cells in the body. This is not easily possible with somatic cell therapies. But of course, there are really important questions about what would be deemed acceptable or not, and there are possible unintended short and long-term effects of gene editing, both for the individual as well as society as a whole. And so I do actually support the call by many others, including this Academy, the World Health Organization, the Wellcome, and the US National Institute of Health, which have really said very clearly that we need to discuss these issues very widely and very deeply to inform on the use of germline gene editing for therapeutic uses, as well as really engage the public so they can begin to grapple with these issues in an informed and rational way. And I think this public engagement will be very difficult because the issues themselves are very challenging to communicate. So in the meantime, while this discussion and consultation process takes place, I think it is important that we do continue to use genome editing both in somatic and germline human cells for research purposes, as it is this that will give scientists and doctors the best chance to make the kind of transformative medicines that we've been talking about today to help patients with genetic and rare diseases. Thanks.
So it's quite useful following on after Matt, because I too don't have any slides. And I just wanted to kind of set the scene. So Matt was talking about CRISPR gene editing from an academic point of view, and he raised one of the areas in which my company that I work for, which is Horizon Discovery, which is a contract research organisation. So essentially we undertake projects for large pharma and biotech, and we use CRISPR-Cas gene editing for a variety of different applications. I think the most important area that we've seen grow recently, which was the area that Matt alluded to, which is where you can, in one go, using one step, knock out all of the genes in a specific cancer cell line population, and then ask the question, how does that affect how a drug works? And that's an important point because we've known over recent years, especially in the cancer area, there are a lot of drugs that go into preclinical trials and clinical trials, and a lot of them fail. And that's a very, very costly process. What we're starting to see with the ability to run these kinds of screens in cancer cell lines is that this is quite a robust way of screening how drugs work and understanding how drugs work. And so the data that we produce from those types of screens are looking more robust to base the initial target discovery for new therapeutics. And that's one of the areas that we've seen grow hugely within the contract research organisation, providing allied technologies to the large pharma and biotech community as we do. Now, what's interesting is that obviously there are caveats to using established cells to look at these kinds of screens. And as Matt's already told you, CRISPR is actually quite easy to use. And so one of the areas that it's allowing development to grow in is to moving away from these established cancer cell lines and immortalized lines, which we've all been working with for very many years, and moving into new areas of research, working with primary cells. So, for example, some of the R&D work that we oversaw at Horizon has been in introducing these kinds of CRISPR-Cas screens into primary T cells. And that means that we can actually address questions using primary T cells from blood donors to say, how does losing this gene or losing that gene impact the ability of those cells to survive? And coming back to what we heard from Dr. Leonard, how does that impact the ability of those T cells to work in an immune suppressed environment in a solid tumour, for example? And being able to work in primary cells or cells that are very close to primary, such as induced pluripotent stem cells, and differentiating those, allows us to take one step closer to the clinic and away from the established cancer cell lines to try and get insight into those early preclinical development roles about how these particular cells are actually going to work. So that really allows us to perhaps understand how new drugs might impact a cell that's much closer to those that actually exist in a human patient, and moving away from some of the more established in vitro models that we've used over the years. I think it's interesting that we can use CRISPR itself in lots of different ways, as Matt also suggested, which is to actually not even have to edit a gene, but to actually use this same system to shut down gene expression, or indeed to overexpress gene expression. And again, the reasons why that's such a powerful technique in terms of research and development, especially within the drug development pipeline, is that you are changing the genes as they actually exist in that cell. And that allows you to understand how that gene network influences the different genes that work in that cell and get a better understanding of the underlying biology. And the more you can understand the underlying biology, the better you can then frame drug development or biologicals or development of antibodies or something along those lines. We are not a therapeutic company, but we do support the work that Dr. Leonard spoke about in terms of we can help large pharma and biotech to look at the therapeutics they want to develop, but also to help them look at the on and off target effects. So CRISPR can be used as essentially a drug therapy, but it is the same as any drug that we take into the clinic. It has good effects, which is what we want, but it also has off-target effects. So we spend a lot of time in our own R&D work also developing areas to try and understand what those off-target effects are and how they can be mitigated to make this a much more safer use. And what's interesting in that context is that we have seen a resurgence of interest moving away again from editing the genome and bringing back into some, one might call slightly more old-fashioned techniques, and that's bringing shRNA back, which again is a clinically approved technique, but given some technologies' worries over the use of CRISPR to actually cut the genome or edit a gene, some technologies are coming back into fashion, so using different and old established technologies to achieve, in some aspects, the same effect. So it's interesting from a research organisation to see how the CRISPR field is driving research and how people are looking at this as it goes forward. Thank you.
So my name's Alison Kay, and I just want to say thank you for inviting me here today to represent the patient view for the neuromuscular community. I'm going to do that by telling you a little bit about my family's story and contextualising that within the Charity Muscular Dystrophy UK, which has been very much our lifeline. So this is my son Bertie, who has Ullrich's congenital muscular dystrophy. So that's a child-onset muscle weakness condition. Now, you've probably heard of Duchenne, but you may not have heard of Ullrich's. Now, this is a collagen 6 myopathy, and it... What was that? And it sits on a spectrum. And the severity can range on this spectrum very much, but also within each individual. So the phenotype can be very different. And that's one of the questions I have sometimes in thinking about what sound to me like amazing therapies like CRISPR. How will it work on an individual level when individuals manifest very differently? We don't always fully understand the complexity of how different genes work together. And even though we may have identified a particular pathogenic variant, it doesn't mean there's something else in there that is part of the story that just hasn't been picked up. We very much went on the diagnostic odyssey that Dr. Leonard mentioned earlier at the beginning of his speech. It took a very long time to get a diagnosis. These conditions are very rare, they're very complicated, and it's not as simple as just ordering a genetic test. You often have to wait for a special study to be happening somewhere. So Bertie is one of the 70,000 people or so, and I say also because we're increasingly thinking that number may not be accurate and there may be many more people. This is partly because people are living longer through better health interventions and non-invasive ventilation in the evenings and things like that, but also because we're getting better at diagnosing. Muscular dystrophy is an umbrella term, and so one of the comments earlier in the Q&A was really resonating with me because there are 60 or so different types of muscle weakness condition. Some are extremely rare, and the present commercial model, I don't see as the mother of a patient how that would work, how people would be incentivized. So I hope there will indeed be a trial to treatment route for my son. But if not for him, then I hope that people can work towards different models of providing therapies for people with rare conditions that are indeed very rare. There is presently no cure. So the neuromuscular community is a very motivated audience watching people quite closely, actually. It's easy to become a nerd about genetics when you have a vested interest in it in terms of who you love. But the neuromuscular community has also experienced quite a few false starts along the way. And I think false starts are great if they take us somewhere and we learn something. But I would say we are cautiously optimistic. It's probably worth explaining that it isn't, when we think about muscle weakness, we often think about mobility issues, but actually people with muscle weakness conditions can have cardiac and respiratory issues as well. There are some links with autism, skin issues, particularly in Ulrich's congenital, and diet and weight issues, all of which can lead to other health issues. So muscle weakness conditions affect people in a myriad of ways. This isn't just about movement. My own son is now 8 and he is semi-ambulant. He can't climb the stairs. He could probably walk on a flat surface from here to there, but he can't run around the playground with his friends or play in the same ways as his older siblings do. But he has a de novo mutation. And I think what's really surprised me is that in many conditions, there's as many as 60% of cases seem to be de novo. And I think that's part of a conversation I don't hear, that although it's now heritable in him, he didn't necessarily inherit it himself. And I think that's something that perhaps I would like to hear more about, but just as a patient, it just doesn't seem to be raised. One of the things that people do when they're highly motivated to find cures is engage in campaigning and fundraising. And I can appreciate that in the wider global scheme of fundraising, there are bigger sums of money floating around. But if I could just say that significant portions of this money are raised by people like our friend Andy here, who ran the London Marathon and is now engaged in a red run where he runs every single day for a year, I could perhaps just bring it home that this money, a lot of this money is raised by very real people with very busy lives who are just desperately trying to fund change, fund therapies and fund cures. And just make that a real journey for people. I think it's easy to become sort of quite distant and quite abstract. And also, as part of my efforts to support the charity, I sit on the lay research panel, which assesses the lay applications that come in after the grant call. And so I've had the great pleasure of reading about a great many interesting projects since I joined that panel. And I wish we could fund them all, because they do sound brilliant. And I also sit on the Content Advisory Group, which is a wonderful group of people who assist the charity to think about issues such as how best do we represent the interests of the neuromuscular community, but how also do we communicate to them about quite complicated issues. Scientific research and things like CRISPR are very complicated for people who are non-scientists and not lab trained, and there are very different levels of understanding out there. I think it's dropped a slide, actually. Oh, it added one in the middle. There we go. So I'm just going to leave you with this, which is, I reached out before I came here to have conversations with people on the different panels that I sit on and ask them about their understanding of CRISPR, whether they had heard of it at all and what they thought. And I think I could summarise briefly their comments in this area. I think there's a massive gap in understanding. I think people conflate the somatic with the germline, and that can lead to misunderstandings, particularly in terms of ethics. I think there are some misunderstandings about whether things will be a therapy or be curative. There's a lot of talk of cures sometimes. When you're needing hope, it's easy to let the pony out the stable and run with it very quickly. But actually, I would like to say that if you have just a very modest increase in mobility or respiratory function, that can be absolutely life-changing. So even if these therapies do not end up as cures, they could still transform many people's lives. People are very unclear about timescale. I think clarity is needed there. And they are concerned about accessibility. And I think probably one of the reasons for that is when we have seen drug therapies prove safe, they have then proved inaccessible for cost means or because regulatory authorities have taken a long time to approve them. There was a very big and ultimately successful campaign for Translarna for Duchenne. And I think people, that's fresh in people's memories. And I think people also could be said to have some concerns about accountability. Who will be the decision makers? Who will decide who has access? And which conditions, when they're extremely rare, sometimes the figure of 1 in a million for my son is quoted, who will make the decision that his cure is worth, or his therapy is worth researching? So thank you for your time.
So thank you very much to the Academy for the invitation to be here, to Dr. Leonard for his fascinating lecture, and my fellow panellists for their very thought-provoking comments. It's a real privilege to be here to discuss this very important topic. The ethics of gene therapy or genetic modification are actually something that we've been talking about for a very long time, so over 40 years, in fact, since the recombinant DNA era. And what's new with genome editing, as we've heard, is that the power of these new techniques to make changes more precisely, more accurately, at high efficiency, and in a broad range of target genes and cell types has meant that there's a hugely widened potential scope of application for genome editing. Now, this has many ethical implications. It means the possibility of treatments for new disease types. It means we can potentially reduce the cost of research and therapies, bringing personalised treatments within closer reach. The fact that these techniques are simpler and lower resource in many ways means easier access to technologies, and this means a wider user base. Now, this has implications for global science and society. It will enable the use of genome editing technologies in lower resource settings, so that affects where people are going to be able to carry out these procedures. But it will also change who can use it. So to give you an example, the way that we used to do genetic modification, so think about knockout mouse technology, for example. There is no way you could make a knockout mouse in your basement, but I can go online and buy a DIY CRISPR kit for $159. Amongst the wider range of human applications that genome editing has opened up, one of the most controversial, of course, is that of heritable or germline genome editing. And as the events of last year showed, it's no longer a question of whether we're going to do it. So as I'm sure everybody here knows, last November it was announced that a scientist had made the first genome-edited human babies. So it's not a question of whether that horse has left the stable. But what we do need to figure out is when, how, where, by whom, on whom, for what reasons we might want to carry out this procedure. And very importantly, who gets to decide the answers to those questions about when, how, with whom, etc. And so I thought Professor Williamson's question here was the one that we're really going to have to face pretty soon, if not already. So if I have embryos and I want to have a go with this technology and I can find a clinician scientist who's willing to do it, should I be allowed to have a go at that? Should I be allowed to have human heritable genome editing for my children? If not, why not? And what can we do about it? Now, I'm going to have a go at answering, hopefully partly answering that question, because I'm going to argue that the most important ethical issues we now face in realising the promise of human genome editing are now about the global governance of science and the science-society relationship. Part of governance is thinking about how we understand risk and determining what is a responsible approach to assessing and managing the risks that will be associated with genome editing, whether it be somatic or heritable. And as Dr. Leonard pointed out, what constitutes acceptable risk may be disease relative. So the more severe a disease, the more a risk may be worth taking in order to try to cure it. So this will have ethical implications for the distribution of the burdens and benefits of research participation, particularly in the case of rare diseases and of small patient cohorts. We're not talking just about risk, but also about uncertainty. That is to say, we're dealing with unknowns. We don't know the effects both short-term and long-term, of some of the genetic changes we may make. And this is again where genome editing is a game changer. Up until the last few years, we were able to say, regardless of whether we thought in theory it should be done, in practice, human heritable genetic modification was too risky. And genome editing's changed that. Now technology and social demand are pushing us to say, well, okay, but exactly how much risk is too much? When and how will we know it's safe enough? And so I think to this end, the moves by the academies and by expert bodies, as Professor Lechner mentioned, to set out a scientific approach to demonstrating safety, to assessing risk, as in fact we did in this country for the legalization of mitochondrial replacement therapy. A scientific approach to that is absolutely essential, and so that's a very positive step. Another point is that we shouldn't automatically assume that somatic genome editing is going to be safer than heritable genome editing. That's often the default position that we seem to adopt. But in fact, there are different kinds of risk involved. There are different people who may be exposed to risk, including future persons who may or may not even exist, depending on the decisions we make. But it's not a given that somatic is safe and germline is not. Now, Dr. Leonard also talked about fixing the broken gene, which I suppose also calls to mind the phrase, if it ain't broke, don't fix it. But what sorts of genes count as broken? What sorts of conditions should we be looking to fix? Again, a risk-benefit analysis will obviously come into this to some extent, so fixing a very serious, life-threatening or life-limiting disease may justify incurring more risk. But what exactly do we mean by serious? So here we get into the question about potential genetic enhancement. At what point does something stop being a disease and start being something that is optional to fix, something that isn't serious enough? I will point out on this, so these are difficult questions with no clear answers, but we do need to account for and be sensitive to value pluralism and the effect of sociocultural context on our assessment of these questions with respect to risk, harm, what counts as a benefit, what counts as a disease. So just to illustrate that, the experiment last year was criticised for many reasons, and I think it was very worthwhile of criticism. But one of the arguments that was raised, so the editing that was performed on the twins who were born last year was to try to make them immune to infection by HIV. And so, of course, people pointed out that there are many other ways than genome editing to avoid becoming infected with HIV, and even if you are infected, it's no longer necessarily a life sentence, it is treatable, it's manageable, etc. And Let me be clear that I think that the work that was done was unethical in many, many ways, but it did stick with me that one of the justifications that He Jiankui gave for the work was to say, well, actually in China it's not as simple as saying HIV is avoidable, it's treatable, it's not a significant condition, because some of the social stigma associated with having that disease in that country might make it more of a life-limiting condition than we might simply think medically. So I just want to point out that we need to take sociocultural context into account. And of course we might say the solution is not to fix it genetically, the solution is to fix stigma. But I will point out that that's also the argument that's raised by many people saying, hey, why are we attempting to eliminate this condition via genetic technologies when what we should be doing is promoting acceptance, accessible society, valuing diversity, etc. Okay, we also need to think more seriously about who might be harmed by genome editing. So again, in the case that was carried out last year, it's not yet clear what the effects have been for those twin girls. They may be perfectly healthy even if they are not immune to HIV. And plus, had the experiment not been carried out, they would not have been born in the first place. So it's hard to say they've been directly harmed. But again, as Dr. Leonard pointed out, this heritable genome editing area is not where the majority of therapeutic work's being carried out. It's not where the majority of the benefits are being or are going to be realised in the near future. And so what we can say is that if reckless, irresponsible and too hasty use of genome editing technologies, particularly in the heritable space, results in public or regulatory backlash, if it results in uncertainty or hesitation on the part of investors, and if that in turn delays the development of legitimate and effective therapies, it's those patients who will have benefited sooner and instead miss out. They will be harmed. And for that reason, responsible and socially robust science is even more important. And that has to include reliable governance and publicly trustworthy science. So this is part of my answer to that question about if not, why not? If you want to do it, why not have a go? Because it's not just a matter of individual decision making. It's about the collective social governance of what is acceptable, what's considered responsible. He Jiankui, in doing what he did, in going against scientific consensus, in flagrantly preempting public discourse, undermined trust and scientific responsibility, and that can have harmful effects on science. We also need to be aware of the importance of responsible discourse about genome editing. So if we focus the conversation on the far future hypothetical scenarios, if we give the impression that the science is all about creating designer babies and superhumans, we risk misleading publics as to the reality of the science and what it can achieve in the near present. And so this brings me to my final point about the role of publics with respect to the governance of science and with the process of science and innovation itself. I won't say too much on that latter, except again to point out that if people are buying DIY CRISPR kits for $159 under the banner of democratizing science, I think what that demonstrates is that publics are unwilling just to be passive patients and research subjects. They're seeking a more active role in participation. Experience with other areas of biomedical innovation and health technology, especially stem cells, shows us that the combined forces of patient need, commercial marketing and consumer purchasing power can significantly distort established pathways for research and innovation and require new regulatory approaches. So the other half of that question, if not, why not, what can we do about it? If there's enough drive to use these technologies, merely prohibiting it at government level may not be enough to stop it. And finally, the role of publics then in the governance of science. There have been numerous consultation statements, expert reports and policy recommendations, and as Dr. Garnett alluded to, a consistent feature in all of these has been calls for public engagement, that we need a broad societal consensus before we move ahead with germline editing technologies. But what is this and how is it to be achieved? Here what I want to highlight is the global dimensions of this issue. So in this country, in the UK, we have a well-established, reasonably effective model of public engagement and of bioethical policy making established really since the Warnock Committee in the 80s. So we have that at national level. What we haven't yet established is how that can translate to international level. And here I think is one of the most important opportunities for leadership, actually, not just in the science and technology, but in ethics, regulation and governance. So our current system for regulating embryo research, genetic modification of embryos, etc., is seen as being an international gold standard. I was in Taiwan recently talking about genome editing, and there were many questions about, well, but how and why does your regulation work, and how can we achieve that sort of standard? So it is perceived as being one of the best models. But it's not as simple as just rolling out our model on a global scale. So what we need to really ask ourselves is, what is that, the current regulatory model that we have here, what is that representing? How is that mediating that delicate relationship between science and society? What does broad societal consensus at a global level look like, and how do we get there? And that's the next challenge we need to address. Thank you.
Okay, well, can I first of all thank our 4 short talk speakers for being very disciplined over time and sharing a very helpful range of views on different dimensions of this topic. So we've now got ample time for further discussion. And again, please do raise your hand so we can get a microphone to you at any point. But if I could perhaps just try to shape the discussion, responding to the themes raised by each speaker. So Matthew raised the interesting point about polygenic diseases, and I'd be particularly interested if John has anything to add on this. So naturally the focus and the logical focus and the obvious application is going to be on monogenic diseases because the case is so much clear-cut and the biology is relatively well understood, etc. But there are many diseases that are polygenic, and I wonder if any experiments have been done to see whether there is some of those genes are more influential than others, and you can actually modify the disease course, picking up the point that Alison made, that actually you don't necessarily need to cure something to have a benefit. Has that been...
That's not work that we're currently doing. It is something we've talked about, but I view this very much as a stepwise process, and the examples that I gave were, I think, illustrative of some of those very first steps. So again, back to those categorical edits, we want to get those things right. We want to be able to understand how with some of the questions about guides behave and make sure that they behave well, because as you add complexity, it gets a little bit more difficult. But I don't know why over time this is something that we absolutely won't be able to address. Yep.
Do you want to add anything, Matthew, to what you said?
Just to emphasize that actually, most of the common diseases that we think can affect people are polygenic diseases, and that the changes that we see in the DNA of people with those diseases often don't occur in genes. They occur in the space between genes that are often very poorly understood. So it is a really big fundamental challenge when you see that change, just figuring out what it does before even changing it, because you don't even know what to measure. So it is a big problem.
Could I add a comment to that? Because it's a really important point. It's the most obvious thing to do is to pursue what we know. And, there's a lot of information about particular genes, but these non-coding sequences have great influence over how genes are expressed and how they interact. And the CRISPR technology and related, sorts of technologies, I think, can be explored to behave or to understand better how genes, in fact, do behave and how they control. And so it's one of the things we had talked about very, very early at our company, which is, could you imagine a screening process where you're looking at actually removing some of these non-coding sequences to measure cellular behavior? And when you identify them, in some respects, you already have the therapy. Because if you have a favorable response and it's as a result of removing something, identifying those guides that, detected in the first place could actually potentially be deployed. So that's work yet to do. It's not something that we're going to work on in the near future, but there's certainly people thinking that way.
And could I just add the role of environment as well, which just I think would be worthwhile mentioning with all these, polygenic common diseases.
So I'm going to steer this discussion through my response to the 4 talks, and then we'll have a completely free-for-all. But anything on that on the issues that Matthew raised that you want to come back on before we move on? Yeah, Lizzie.
It's another really speculative question, but the effects of stress will change the epigenetic marks of your DNA for the generation below and the generation below you, and who knows after that. I mean, that's well established. So do you envisage that you might be able to do something like treat PTSD using CRISPR-guided changes?
I'm going to claim a bit of naivety about that. I mean, I think certainly with CRISPR, we have the tools to make epigenetic changes to the DNA and have the tools to study that and understand how those might relate to stress-associated diseases. And presumably then the CRISPR therapeutic toolkit, once you understood that adequately well, could be expanded into that space if it was certainly a monogenic disease. Again, you may be in the space of kind of polygenic disease as well. But I think, as was mentioned by John earlier, when one looks over the fullness of 20 years' time, I don't see any fundamental constraint except of our lack of current understanding.
Okay, so then Nicola highlighted the value of this kind of technology in relation to drug targeting and identifying genes, if I understood you, that would be safely, if there was an off-target drug effect, you can identify genes that don't matter. It reminded me, Richard Trembath I saw slipped out, but he's been engaged with a very interesting program called East London Genes and Health. I don't know if anybody here is associated with it, where they've been studying a population in the Tower Hamlets where there's an unusually high incidence of consanguineous marriages. And so studying this population at the genomic level, they've uncovered quite a large number of human gene knockouts, many of which are silent. So it's just illustrating almost how many redundant genes we have, which again is very useful in terms of drug development, because it's highlighting genes that if you have an off-target effect that affects this particular pathway, it probably doesn't matter. But that was the sort of thing I think you were hinting at.
Yeah, it was more around the fact that you now have the capability to take a cell population and knock out all of the known coding genes in that population and then use that to ask specific questions. So yes, it's from that point of view, it's quite a powerful tool set to be able to work in that arena, which I don't think we've been able to do as effectively and as efficiently and as rapidly as before. So similar to some of the points that Matt was making. I think it's a very powerful arena within finding new therapies, whether that's in the drug development space, but whether it's also finding how specific genes interact indeed, and it might be that you can target one and not the other, or that there are specific benefits from targeting a particular gene in a particular population for a particular disease. And it's really how CRISPR allows us to address that through its many different manifestations. And I think those manifestations are going to grow over time. So obviously, the other area of drug discovery that we know has an impact is how, as a population, people respond to different drugs. Anesthetics is a known area. People can respond very poorly to certain anesthetics, and that's due to pinpoint changes within their genome. And it might be that as CRISPR evolves, indeed, I suspect as it does evolve, you'll be able to ask those questions on a cell basis instead of a patient population basis. So you can address those in a dish, not as a risk in the theatre, for example.
Yeah, Janet.
might be a very naive question, so forgive me. But not all DNA in the cells are the same. We're talking as though all the DNA is naked DNA, equally accessible by CRISPR-Cas9. Yet we know large proportions of the genome are packed very tightly. So when you're doing this type of screening, how do you overcome, is there a bias, and how do you then overcome the bias to being parts of the active genome?
That's a very important question, and I don't really think that we have a particular handle on that to be able to answer that yet. And I think with the CRISPR community, there are lots of open source data. And so I think that that's very much going to be, as we increase the number of lines that are investigated through CRISPR screening, and that information is made available to the public, there are certainly companies already in existence and academic labs already in existence that are taking those whole data sets and then using them to mine them to actually understand, well, you know, did gene X in this particular cell type knock out properly or overexpress properly or knock down properly versus this particular. And we are generating those gene set data to be able to do that, but I don't think we are able to do that currently. But it's a valid point.
Matthew.
I think it's a fascinating question, and we've done quite a lot of screens where we've knocked out every gene in the genome, and both genes that are actively expressed and genes that are repressed, and look to see if we can see patterns about why CRISPRs work or don't work. And we've not seen much. So we've asked, for example, does the pattern of methylation within genes fundamentally distinguish which CRISPRs will cut and those that don't cut? And we don't see that signal. I don't think we can totally exclude that there are other factors, but actually, I think quite surprisingly, it does seem to cut DNA that is probably quite compacted in large, tertiary structures. So it's quite remarkable. One thing we do know is that CRISPRs introduce DNA damage when they cut cells, or they cut the DNA in cells, and genes that are amplified, particularly in cancer cells, or if you cut those, will lead to more non-selective DNA damage and are more toxic to cells.
Okay, so then, Alison, I thought it was so valuable to get the perspective from well, you're not a patient, but someone very closely connected with a patient. I thought your remarks were all really insightful, and I think the point about modest gain of function can be transformative. It's a really well-made point. The other thing I thought, the other issue you raised, which is important, I'd be interested if John's got any comment on this, is that single gene disorders can have quite different phenotypes. And that's not surprising, really, because there are so many other genes that may impact on the function in question. And to what extent is that something that you've thought about with the diseases that you're going after?
Yeah, I mean, it's true. When we think about using CRISPR as therapy, in the examples that I talked about, these would be patients who we can identify who actually have manifestations of the disease. So I think we'd have a good sense that something's happening, and if there's a good link to that underlying gene, we would feel reasonably confident acting on it. If there's modifiers beyond that, that's going to be harder to deal with, and that's just a fact and a limitation of the knowledge that we have. But it does start with having an understanding of what the primary actor actually is, and I think one can usually get a pretty good sense of that degree of contribution. So it's not a mixed bag, and you don't know if it's one thing doing 10% of the effect, for example.
Yeah, I mean, a risk, I'm sure, of being misled by working in recombinant mouse strains where they're genetically identical, unlike the messy human species we are. But I guess maybe it's reasonable to predict that if you do deliver gene editing to a population of patients, you might see some variation in response. I mean, that would be a reasonable prediction to make on the basis that their background genes are different. And then moving on to the minefield of ethics and all the issues that you raised, Sarah, which are so interesting and almost inexhaustible. I'm not really quite sure where to start. I think that I was glad that you highlighted the fact, as I was saying to John and other colleagues from Intellia over lunch, I do think the UK is particularly good at regulation in an intelligent, public engaged way. And indeed, so much so that I hope if one's looking for any kind of silver lining in relation to the pickle that we're in at the moment, that maybe we can market ourselves as the go-to place for innovative therapies in this kind of territory because we are good at regulating them. I would like to think that that's the case, and we'll persuade our Intellia colleagues that that is also the case. One of the things that crossed my mind is particularly if one's thinking about germline, the response to GM crops. And some of your colleagues, prominent colleagues, I think, in the US have adopted some interesting attitudes to this, as have some of our European colleagues too. So what have we got to learn from GM crops when it comes to this technology in humans?
I think one of the important lessons is actually less about technology itself and more about how we manage the science-society relationship. So the GM crop case has often been heavily criticised for not involving public soon enough, for not being sufficiently engaged and responsive to concerns, for trying to railroad opinion and sweep things under the carpet. I'm pleased to say that from what I'm seeing so far in the human genome editing space, that's not going to be the case. So there has, as I say, from very early on, the view has been expressed repeatedly that what we need to do is engage publics on this to understand not just whether or not we should go ahead with the technology, but actually what sorts of uses are and aren't acceptable, what are the needs, the desires, the challenges, the hopes. One thing that I think is very interesting, so I was involved with some work that the Royal Society did on this, the public dialogue that took place in the last couple of years. And what we're seeing is that attitudes towards human genome editing it's definitely not all about the public being scared or concerned or worried about these technologies. There's a lot of hope out there, there's a lot of enthusiasm, there is a perception that there can be this huge benefit, and what people are really concerned about is how do we shape this, how do we go forward with it to make the kind of society that we want to create. So I think that's really encouraging.
Anything you want to say from a US perspective?
Well, you mentioned colleagues. I was wondering which you had in mind before I
was thinking of some of your Republican. They're not colleagues. I don't mean compatriots.
Compatriots. Okay. Well, there's 330 million of them, I guess.
Some who make more noise than others.
Yeah. I mean, I agree with what's been said, and I tried to only make the point at the end that there's incredible complexity here, and this is, changing the food we eat, the environment we live in, and to the extent that, you go beyond, you think of ecosystems and monoculture kinds of, crop sources. It's not just about the traits that one introduces into the plant. It's all of the knock-on effects. And, we have to decide. But I think those decisions are best done after there's a full vetting of as much information as we possibly can have. And I welcome that, I support that. And as a consumer of, food and animals and all that stuff, I think to do otherwise would be a terrible, terrible error. Yep.
There are a couple of questions. Yeah, one here and one over here.
Robert, if I may, I have a quick question to expose my ignorance and then a follow-up. So the quick question to expose my ignorance is, are mitochondrial transfer considered somatic or germline?
That's not. So that doesn't expose ignorance at all. It actually exposes. It's a very complex situation. It's a good question. Okay, so what we've been calling mitochondrial replacement therapy will affect the genes that are present within the next generation. For that reason, and it will affect their children's genes, etc. And so for that reason, people have argued that it should be considered a germline modification. However, we're talking about a different combination of mitochondrial and nuclear DNA rather than making a DNA sequence change. So I suppose we need again to think about what's the germline somatic distinction doing for us here. I argued already that it's not as simple as saying one's risky and the other is not. So I would say mitochondrial replacement therapy and nuclear genome editing are different kinds of things, and we need to assess the risks of each of these for what they are. In respect to what's legal and what's not, we have a very specific situation in the UK, which means, in general, you cannot have heritable genetic modification, but mitochondrial transfer is an exception. Does that somewhat answer the question?
It does, because I'm Steve Bates. I run the UK Bioindustry Association, and I was interested in the recent ARM, Alliance for Regenerative Medicine, position statement that went to the World Health Organization, which was signed by Intellia and a number of other US members of the alliance, which did make a clear distinction between somatic and asked for, and made the case that, and germline, and made the case that there wasn't yet the case for germline editing, whereas I think in the UK, in a sense, we're already a step beyond that because of the mitochondrial transfer work that's gone through Parliament. It's to your point, Robert, that the UK has been very good at this. If I was going to characterise this debate in the UK, I would say when this science has developed, the UK has put a lot of money and effort into getting the rules and the ethics together, and the Americans have done very well at getting some venture capital-backed companies up and running, and we're now at the place where there's quite a lot of US companies, but the US companies are now thinking, actually, we are nervous or concerned about the limits to where we've got to. And my nervousness is whether seeking to take that approach to a global discussion, we've had the scientific discussion, we've obviously had the experience in China, we may have had the experience in other parts of the world. Of course, there was mitochondrial transfer, I believe, in Mexico first, done by Americans who moved outside of the US in order to be able to do the work. I think my point here is, if we're going to develop this as an industry, and I take the caveats about very rare diseases being very difficult in terms of business models, are we at risk that we have a nervousness from American invested companies? And this goes to the point about GM food. We of course had genetically modified tomatoes on sale in the UK before they were banned because people didn't like the way that Monsanto was seen to be operating as a capitalist enterprise, which we have to put capitalism into the discussion about science and society, which is different in the UK in terms of this debate because we have an NHS and everybody is automatically enrolled in healthcare before any decisions are made. And therefore the translation of this from a national context to a global context, which I think both Intellia have been sensitive to and engaged in, and you see the discussion that the Academy's been involved in, is actually a very tricky one. And you were right to point that in China, it was interesting for me, I think that they had a discussion about, was this person a national hero or a very naughty boy? And it took the Chinese Communist Party a little while to come to the decision that he was actually a very naughty boy. And how we get to a discussion, which is how do we appropriately move the space for this, the regulated space for this forward in a way that's both appropriate with the science and appropriate with the business model and appropriate with the society is what we're trying to do, and it's actually quite tricky, so I think we need to work together on it. And I don't know if John has a view, whether there's a space for the UK to play a role, as Robert suspected, in this as a global industry.
We were talking about this at lunch a little bit. I guess you could say, we are a consumer of regulatory input, and in my experience, as we look at our own country, the United States, and some European countries, I would say the MHRA stands out in a very positive way. It's very informed, it's engaged, it's, I think it's very mindful of the medical problem that's trying to be solved. And I think, you know, as one thinks about the practice of medicine and public health, that is the proper stance for a regulator to take. And, you know, we're actively interacting with them and hope to continue to do so. Don't lose that. It's very important. And I really do believe that the UK is a leader based on what I've seen so far. But, you know, there's in the end, what we do, we want people to trust and believe that the information is correct, right? And that is something I think that needs to be held fairly sacred, because people make decisions based on, you know, the information that we present to them. So do the experiment and do it properly and have results that are interpretable and foster that. Continue doing that, but do not let this become like, and I think there's no risk for this, by the way, but the example in China that was cited, I think verges on the wild, wild west, and is, I would argue, medical decision-making at its worst. You know, and I understand there's contextual stuff and all that stuff, but I think that is, and you mentioned this in your comments, those are the sorts of things that cause us to throw the baby out with the bathwater, which I think would be a terrible mistake.
Can I comment as well?
Please, Sarah.
So, Steve, I think you've raised a really important point there that's worth just exploring a little bit about the potential effect of transnational regulatory differences. So I alluded to the difficulty of achieving a global discourse, but we also have the very immediate problem of, if we allow it here and not somewhere else, are we going to become the destination? If vice versa, is all our innovation going to go overseas? What I want to point out with respect to that is, it's not just about the hard regulation or about the laws that we have that brings about these tensions. So you referred also to the Mexican, the mitochondria case. So the first actual transfer of a mitochondrially replaced embryo happened in Mexico. And the scientists concerned there said, oh, it's because there are no rules. Now, actually, that's not the case. There are rules. But he was taking advantage of lack of oversight resources to enforce the rules and of a perception of this. In Mexico, I guess it's the Wild West or the Wild South. People have made similar comments about China being the Wild East. And so there are, it's not just regulation, but it's the global dynamics and politics of science that I think we do need to take a bit of a hard look at, to say, when we think of certain countries as being deficient in regulation or with lower ethical standards, or when we cast shade on, well, of course it would happen there, you know, what kind of dynamics, what sort of politics is that playing into? When we come back to the hero-anti-hero narrative, when we put so much emphasis on, well, who was the first? I mean, we may all think he was an anti-hero, but He Jiankui is still going to be in history as the first to do it. What sorts of, you know, the narratives we create, the ways in which we create value in scientific work, how are those also pushing us towards these dubious situations?
Yeah, I mean, a particular complexity comes to mind, and that is, you know, there's talk of gene drives being put into the mosquito populations, for example. This is going to happen in countries which probably just don't have the infrastructure to have the debate. So I think the sort of national permission or societal approval is tricky. It'll be done to rather than done by. John.
Yeah, I too was going to draw parallels with mitochondrial transfer, but Steve has raised many of the issues. I'd just emphasise one point which I think is instructive in terms of how well this went down ultimately in the UK, is the time frame that was involved in securing public engagement and sensible discourse about its use. And I think we need to learn from that and recognise that. And I was delighted to learn the Academy actually is involved in a partnership, international partnership, to try and address some of these broader issues. Because to your point, John, about the trust in science, as science becomes increasingly global in its interpretation, a bad player some way somewhere is going to influence, in a GM crop perspective way, how our society views the outputs of science.
Yeah. Could we come to you later, Bob? This lady here.
Hi, I just wanted to touch again on the point about accessibility that Alison raised. So it strikes me that there's a couple of interesting things about this particular niche. First, on the patient side, and you show the picture of your son, Alison. There are lots of reasons why we as a society may want to prioritise that type of patient for very good reasons. So the fact that it's children, the fact that these diseases are often very severe, they can be very painful, lots of stigma associated, short life expectancy. There are lots of reasons on the patient side why we might want to prioritise treatment. And then on the technology side, this is really cool stuff. It's really innovative. It's genuinely disruptive. And I wonder whether or not those two things sometimes get conglomerated to the point that, are we incentivising the patients or are we incentivising the technology? And what is it that we're willing as a society, or potentially not, to pay for? So if they come with a big price tag, is that because of what they do, or is it because of how they do it? And does it matter? And that's a really big question, but I wondered if anyone wanted to have a go.
I'll leave Alison to reflect on that one for a moment. That's a tough one. And of course, I don't know who you are, I apologise for that, but you probably know the data that looks at the amount of funding for research in certain areas compared to disease burden. And cancer is the big winner. Mental health is the big loser. And I think that, for all sorts of reasons, I mean, partly because one of John's key criteria was tractability. I hope mental health issues are getting more tractable, but they're still jolly tough. But I think there's also a stigma. There's all sorts of reasons behind this, and I think I'm glad to say there's a big surge of interest in boosting mental health research funding. I doubt many of those mental health conditions will be dealt with by gene editing, but who knows? But it's a really important point. Sarah, do you want to add anything on that?
I wanted to try and identify at least two important points that came out of your comment. One is about the perception of the technology and the excitement, and we have, again, in other areas, most notably stem cells, seen this kind of hope and hype cycle that it may drive up prices, although it doesn't necessarily, but what it does do is drive demand and enthusiasm. And on the one hand, that's great because there's support for therapies that we want to come out, but it can become overhyped, and then that can lead to exploitation of patients, etc. So, there are both good and bad aspects to that. The other one was about the cost of therapies, and as I think has been already pointed out, gene therapies may have to have a very different cost model because it's the one-off and then hopefully you're cured. Some of you may have seen in the media recently, there was a lot of criticism of a particular pharmaceutical company because it was reported from one of their, I think, internal discussions that they'd had a report about genetic therapies and said, oh, this is going to be not cost effective precisely because it'll be that one shot. And so there was a lot of criticism about, well, this just shows that pharmaceutical companies are only in it for the profit. What the headlines traduced and did not go on and report was that they said, because we can't use the same cost model to realise these therapies, we'll have to do it in a different way. And I think that's entirely reasonable. We have to say it isn't just about the profits, but if it's going to be not just about the profits, what mechanisms can we have, what broader social and policy incentives are there to make sure we get the therapies we want and need?
Great. There was two questions over here.
It's easy. So one can readily understand how the delivery mechanisms, which are generic to different categories of disease, can be optimized, and so patient safety will result from other people having had safe treatment before. And the bit which, of course, would be different, particularly for the rare diseases, would be the new guide RNAs which are being used. And so in Dr. Leonard's talk, he mentioned about making general rules for getting safer guide RNAs. I wondered if you could very briefly say any more about that. It seems to me that if the kind of core problem, as we heard in the individual case described, was that if you have a completely novel target, and there's only a few people with that condition, it's always going to be the case that for those rare conditions, there won't have been patients going before having the relevant guide RNAs. Could you say any more about whether it really is just down to trying guide RNAs, or if there are general principles of safe design?
Yeah, thanks for the question. I would draw a distinction between quote unquote safe RNAs and extrapolating data from one disease condition to another. And both are important, but they're somewhat different. So there was a question earlier about, is it perfect? No, but we try to get as close as possible to that by having guide RNAs that we can characterize empirically and bioinformatically that will do what we want them to do. And we measure that, right? So, and what does that mean? That means they cut at the place that we want them to cut. And then there's, you choose the edit you want to make as a result of that. Extrapolating what we learn about efficacy and maybe more importantly safety from one disease entity to that we study and develop some level of confidence on to some other disease state for which there's many fewer patients is an exercise, again, in benefit risk. It keeps coming back to that. And the point I was trying to make is that if the amount of data that we can collect in humans is tiny by virtue of it being a very small patient population, and some of these considerations about cost, which are very real, and just the cost of development in the usual sort of model that we have that is, I think, appropriate but expensive, then I only pose the question, if we learn that elements of what we deliver, in the ideal case here, where the delivery is held constant, the molecules are the same, the innards, if you will, of this delivery mechanism are the same, with the exception of 20 nucleotides that we can measure actually how they cut and how they behave in the patient's own cells before we administer it. Is that a way that we can, depending on the condition, reasonably take that next step of going and building on the safety information that we have from prior patients, certainly with respect to how the delivery vehicle behaves, and take the step of administering that to the next patient with a different disease state? If we don't get to models like that, I think we're going to have some very real barriers that make dealing with whatever is addressable in that long tail inaccessible. Yep.
Yeah, Matthew.
Whenever you make a cut in the DNA of a cell, even with your best predictions, you are likely to get unanticipated consequences at some frequency, I would argue. It may be very low. And I think it goes back to some of the arguments about, benefit versus risk, and I think we can get pretty good at predicting how frequently we might see these unexpected consequences. But it also might, I think quite likely that it'll depend on the context in which you cut. So if you have a region that has a lot of regions of DNA that are highly homologous, that would be perfect for recombination events, for example. Again, we might decide that that's an acceptable risk. I think as well, within that, it may also depend if you're modifying a somatic cell versus a germline cell. Because I think generally we would be willing to have a higher threshold of that risk when you're having something that wouldn't be inherited in future generations than you would if you were modifying the germline, where that would propagate through that individual and that individual's family in the future.
I just want to make one comment. That's correct. I would offer the additional perspective that DNA is frequently represented as this pristine helix with 0 errors in it. And as you look cell by cell by cell, across an organism, there's no variability. It's perfect. That is incorrect. There are multiple double-strand breaks that occur on a daily basis in our somatic cells. And that's why we have DNA repair mechanisms that live within our cells. So if you're looking at a population of lots of cells, and ask, are there DNA, double-strand breaks, primary cells, for example, you'll find them all over the place. I mean, they're there in our cells, within our bodies right now. And the real question is, can you detect any number beyond what's already there? Because we're dealing with randomness on a daily basis. And to the best of everybody's ability, and I don't mean us, our company, but industry, academics, et cetera, the goal is to drive that so that when you look at that population of cells, you can't tell if there's another double-strand break somewhere in there that wasn't there before. Now, if you find hotspots, that's a different issue. And that's what one tries very hard to avoid, and because they're observable. But there's randomness throughout, and that's unavoidable.
Yeah.
Oh, actually, could I just add?
Yeah, sure.
I thought that question was really interesting because it brings me to thinking about when we get there that you will need patient consent and people will need to understand risk. And one of the things that I'm learning in my studies as a genetic counsellor is that people perceive risk very, very differently. And it's actually the bread and butter of what genetic counsellors deal with, because 5% risk to one person means one thing. It means, oh my goodness, I'm not going there. And 5% risk to another means nothing to worry about at all. And I think that's something that might have to be dealt with quite carefully in the future, because depending on how your child is doing, or if you're an adult patient, how you're doing, you might want to take the risk. But another family in the same set of circumstances might not. It was very interesting.
Sarah, yeah.
I wanted to add a comment on Matt's point about somatic versus germline risk. So I tried to argue that actually it's not as simple as one's more risky than the other. I just wanted to elaborate on that and to see what the scientists think of this. The way I look at it is that you might do somatic editing, and you're right, if something goes wrong, that won't be passed on. So you've got a bit of a safety net in that way. But then somatic editing may not be as effective for certain conditions. So it may be better that all of the cells, say, in that tissue are gene corrected from the start than that you're trying to correct it later and then get the modified cells in and colonizing, etc. And then the other thing is, if you do a germline, if you do a heritable edit, and that works, and then you've fixed it in every generation, that's great. Whereas if you do a somatic edit and it works, you then have to keep on fixing it in every generation after that, and that is those procedures themselves entail risk. So if it's, say, if it's one of the hematopoietic diseases, you're taking bone marrow, you're manipulating that, you're having ablation, etc. So these are all procedures that have risk and harm entailed. When you're talking about doing that generation after generation, may there not come a point where actually you think it'd be better just to try the germline edit and fix it once and for all?
Yeah. I'm getting signals that I'm being a bad chairperson and we need to wrap up. I'm going to allow one more question because it's such an interesting discussion because you've got the microphone, and then we'll draw things to a close.
Great. It's Jane from Genetic Alliance UK. It's a really fascinating panel discussion. I'd like just to ask the question of the panel whether they feel that there is actually something missing from the facilitatory regulatory environment for genome editing, because we can learn from history. And if we look back at the early period of gene therapies, We had the Clothier Committee in '89 set out the ethical principles for gene therapies, and subsequently the Gene Therapy Advisory Committee, who advised on the fundamental aspects of ethics, but also performed a horizon scanning function, a public engagement function, and crucially provided oversight on an ongoing basis to gene therapy clinical trials. And of course, in the States, we had the RAC, the Recombinant DNA Advisory Committee. Are we missing something similar here? Because the way I see it at the moment, any human clinical trial of genome editing would simply go through the GAFREC system.
Any comments anybody wants to make on that? So the only contribution I've made is to point out that we have this cross-academy, cross-national boundary working group or consortium looking very specifically at what would need to be the case before people would be likely to approve germline editing. But you're raising a broader question, I think, about this whole field of gene editing technology and whether we have the right things in place. In the UK, I think at the moment one would assume it'll fall somewhere in MHRA GTAC territory, but do we need more?
I think we have enough in terms of the formal regulation and oversight. If there's anything missing from the broader picture of how we do it, I would say that attention to what happens at the global level and how we integrate that, we could look more at that. And then at the local level, it's not that there's formal regulation missing. I think we're doing a good job in terms of continually saying, yes, we must engage with publics. There is, to me, still a piece missing in that we don't 100% know or have a well worked out conceptual model as to when we've engaged with publics, exactly what we then do with that. And that's another sort of piece of the mechanism that I think we could get better at.
Okay. Well, let's draw this to a close. Bob, I'm sorry I didn't come back to you, but over drinks. So I think it's been a fabulous session, and it would be a waste of your time for me to try and sum up, but I'll just make the following points. Firstly, I think this can only reinforce the view that I'm sure many of us brought into this session, that this is an extremely exciting field of medical advance that has enormous potential, and I think I'm very confident it'll have potential beyond monogenic disorders, not only also in sort of modified gene-modified cell therapy, but also perhaps for more complex genetic disorders. Secondly, I think there's every reason to think this is going to yield biological insight. So that's the sort of stuff that Matthew was talking about. The many genes in the human genome, we don't know what they do, and there's all sorts of ways, I think, in which this is going to shed light. Thirdly, that it's crucial that we keep the patients and public absolutely on side and with us, and we have their permission. And we're very fortunate to have expert patients and people like Alison to work with us, and I think that's really crucial. And finally, on the regulatory front, as we've acknowledged, I think it is a national strength. And at this point in our Brexit cycle, we need to be clear about what our national strengths are and capitalize on them. And I think that will definitely apply as we navigate our way through what is an ethical minefield in terms of the opportunities this technology is going to throw up. So those are my closing remarks. Can I thank our speakers, and especially John, for giving us such a great plenary talk. And can I also thank Jim Squires, who manages the forum on behalf of the Academy, and several other Academy staff who are in the room for all the work that went into making this a great success. Thank you very much. And there's refreshment downstairs.